it's Paul. Welcome to FNA Lymph Nodes Case 6. This is an 81-year-old male who had a large submandibular lymph node sampled. This is the air-dried slide, and you can see it's quite interesting. Very busy, lots of nuclei present, and they're sort of dispersed, but also in messy clumps that are really quite kind of tight. Doesn't seem to be a lot of cytoplasm. High power confirms that. You can see really tightly moulded little groups of cells, nuclei that is, some elongate-ish ones as well as round and things in between. You can almost make out places where nuclei meet that there is some moulding. They're sort of wrapping around each other a little. And areas like this, which are almost like linear chains of cells. So you can see the pattern's pretty similar. There are lots of these very tight little groups. I can't really see any cytoplasm yet. So this would be almost what we would call a small round blue cell tumour on histology. So it's an undifferentiated looking malignancy, and it seems to be moulding quite a bit. Higher power, you can see actually there's quite a bit of variation, size and shape of nuclei here. Some quite large ones next to fairly much smaller ones. And some of them are round, some of them are regular. Look at this, chains of cells running down like this, and they're moulding around each other, just like vertebrae in a vertebral column. I can't see any cytoplasm on the cells. There's some very, very tight groups there, some single cells, and this is the alcohol fix slide. Features, so dispersed nuclei and then very tight groups of wrapped up nuclei. There's a bit of crush artifact here as well. High power, interesting chromatin. It's kind of quite fine, but then it has little dots inside it, so we would call that stippled chromatin. Some of them are round, some of them are a little more elongate, and they are in places wrapping around each other a bit. Compare them to the size of the red cells. You can see they're probably 1.5 to 3 times the size of the red cells, so they're fairly small. This area is showing that granular background, that's necrosis, and there are little dark dots. They were apoptotic bodies. You'll see them around everywhere. Tighter groups again showing that similar tight moulding and absence really of any prominent nucleoli. We can see maybe very small ones. So they're what we would call inconspicuous nucleoli. So in summary, we saw a dispersed population of cells, along with some very tight groups suggesting some cohesion. The cells had a very high NC ratio with very little cytoplasm obvious. Nuclei varied in size, but also in shape, from being round to more elongate. There were some areas that showed definite moulding of nuclei around each other and the formation of like vertebral columns of cells. The chromatin was stippled, there was inconspicuous nucleoli present, and the background showed necrosis and apoptotic debris. These features are in keeping with metastatic neuroendocrine carcinoma, and in this site probably the most common primary sites for that would be the skin, and that would be a Merkel cell carcinoma, or the lung, which would be a small cell carcinoma. We would need to exclude lymphoid cells because these cells are quite small and very similar to lymphoid cells. We would do that morphologically, initially by seeing an absence of those lymphoid characteristics I talked about in the last case. So the crescent of cytoplasm, very dispersed population, and the presence of moulding, stippled chromatin, inconspicuous nucleoli, all features favouring against really lymphoma, as is the degree of variation in nuclear size and shape. lymphomas are rarely as pleomorphic as that. We would also use additional techniques to probably confirm that, including immunohistochemistry and possibly